I'm delighted to introduce our next speaker, Fiona Neylon from the University of Limerick, and Fiona is studying industrial biochemistry. So the floor is yours, Fiona.
Thank you. Hi everyone. So my name is Fiona Neylon and I'm a final year industrial biochemistry student at the University of Limerick. So the research I'm going to be presenting today is just a little bit on my FYP, which was titled The Comparative Genomics of Emerging SARS-CoV-2. And my supervisor on this project was Professor Tony Pembroke. So I'm sure many of you are aware, but just a little bit about the emergence of SARS-CoV-2 first. So it actually emerged in December 2019 in Wuhan in China, when there were several cases of pneumonia of unknown origin reported. And then just 1 month later, in January 2020, the source was identified as a novel coronavirus, and preliminary genetic analysis revealed it was actually related to SARS-CoV. So in accordance with this, the ICTV named the new virus SARS-CoV-2. And then just 2 months later, in March 2020, the disease it causes, COVID-19, was declared a pandemic by the WHO. So as of this month, there have been over 120 million cases of COVID-19 reported worldwide, including 227,000 in Ireland. So like all living things, viruses contain genetic material comprised of nucleotides, and then a unique sequence of nucleotides will encode for a particular viral protein. So each triplet of nucleotides, which is also referred to as a codon, will translate a particular amino acid into the final protein product. So SARS-CoV-2 contains 29,903 of these nucleotides, and it encodes 16 main non-structural proteins and 4 main structural proteins. So as the name suggests, the structural proteins give rise to the structure of SARS-CoV-2, and they can be seen, their locations can be seen there in figure 1. So it's important to mention that as the virus spreads, it is acquiring mutations in its genome, and this was basically the foundation for my project. So what I did was I collected local and variant SARS-CoV-2 genomes from the GISAID database, and I compared them to the Wuhan reference genome. So the local sequences included one from each of the Munster counties, and then the variant sequences included one from the UK, South African, Brazilian, and also a mink variant from Denmark. So just the variant sequences that I'm sure many of you have heard of in the media in the recent months. So the comparison was done using a software called MOV, which basically compares genomes by lining them up. So figure 3 just shows a MOV output there. And basically the red breaks in the genome are indicative of discrepancies from the reference genome. So you can see I have a break circled there at position 5986. And that basically just represents what was a C base in the reference genome was actually identified as a T base in the UK variant sequence. So in doing this for each of the individual sequences, I identified 306 nucleotide mutations. So I then put this table together to kind of just summarize in what sequences and in what regions of the genome were each of the nucleotide mutations occurring. So as you can see, the Munster sequences carried significantly lower numbers of mutations in their genome. So the average was calculated as 14.17 nucleotide mutations per each of the Munster sequences. And then the variant sequences carried an average of 44.2 mutations. So obviously the variant sequences carried significantly higher numbers of mutations, which was somewhat expected. And then I just want to bring your attention to the region encoding the spike protein. So this is one of the main structural proteins that I spoke about. And across the board, across all sequences, the region encoding this protein acquired the most mutations. So of the 306 nucleotide mutations identified, 82 of those occurred in the region encoding the spike protein. So using multiple alignments of the resulting amino acid sequences, so the nucleotide sequences were translated into their amino acid sequences, it was determined that 191 of the 306 nucleotide mutations led to amino acid alterations in the main viral proteins. So this was done using software called GeneDoc, and figure 4 just shows an example of how a segment of the S protein multiple alignment was conducted to identify amino acid alterations. So you can see there, for example, at position 27, the core spike protein had a threonine residue, whereas the other spike proteins had an alanine residue. So the reason we're interested in amino acid alterations is because of their varying properties. They can impact protein structure and function, and this could potentially confer resistance to existing therapeutic treatments and vaccines. So alterations in the S protein, or the viral spike, which I've mentioned, are of major interest for two reasons. So firstly, this protein is the protein by which the virus attaches to human cells via the ACE2 receptor. So alterations in the spike protein could enhance ACE2 binding and therefore increase viral transmissibility. And then second, the S protein is also the main vaccine target. So there's 3 vaccines currently licensed for use in Ireland: the Moderna vaccine, the AstraZeneca vaccine, and then Comirnaty, which is the Pfizer vaccine. And all of those target the viral spike protein. So of the 191 amino acid alterations identified, almost 42% of those were occurring in the spike protein. And then if repeat events are discounted, 32 unique S protein alterations were identified. So I then put this diagram together to kind of just highlight in what regions of the S protein were each of the amino acid alterations occurring. So as you can see, there was amino acid alterations occurring right throughout the structure of the S protein. And then just to explain the notation used here, if we take the first amino acid alteration, for example, S12F, that just means that a serine residue, which is a type of amino acid at position 12 in the Wuhan S protein, was identified as a different residue, a phenylalanine residue, in one of the other S proteins. So not all of these S protein alterations have raised concerns over viral pathogenicity or vaccine efficacy. And in fact, only 4 from this analysis are of major concern and were of major interest. So firstly, the D614G mutation, which is located at the interface of the S1 and S2 subunits of the S protein, was of particular interest purely due to its prevalence. It was carried by every sequence that was analysed in the study. And then also there was 3 other alterations which were located in the RBD of the S protein. So alterations in this region are of major interest simply because this is the exact region of ACE2 binding, and therefore alterations in this region could enhance ACE2 binding and therefore increase viral transmissibility. So having identified these amino acid alterations, it was then of interest to model the S proteins in 3D to determine if they were actually having an effect on the protein 3D structure and then ultimately the protein function. So these were then superimposed onto each other, as can be seen in figure 7. And in doing this, a number of structural changes from the Wuhan reference S protein were identified. I'm just going to talk about one of the regions of structural variation that were identified for the purpose of this investigation, or for the purpose of this presentation, sorry. So one region of poor conservation was between amino acids 619 and 632. And it was determined that this was likely caused by the D614G alteration because 3 of the sequences contained this alteration in isolation, but they also contained the poorly conserved region between amino acids 619 and 632. So this structural change, since the D614G alteration involves the loss of a charged residue, the structural change may be explained by the remaining charged residues in the protein attempting to balance themselves. So SARS-CoV-2 harboring G614 is thought to be more transmissible than wild-type SARS-CoV-2, and the structural change identified in the homology models in this analysis may facilitate this by enhancing ACE2 binding or receptor binding. This can't really be confirmed for certain just yet, as other mechanisms by which G614 increase viral transmissibility have also been reported. So ultimately, further study is required to elucidate the exact implications of the D614G alteration, and then also the involvement of the 619 to 632 structural loop, which was identified from the homology models in this study. So to kind of just summarize my main findings and conclusions. So as I mentioned, the variant SARS-CoV-2 strains carried significantly higher numbers of mutations than those circulating in Munster. So the average number of mutations carried were 44.2 and 14.17 respectively. So there was a big difference there. But in all cases and in all sequences, mutations were concentrated in the S gene, so that's the gene encoding the main viral spike protein. So D614G was identified as a particularly common alteration, and it is thought to increase viral transmissibility, although the mechanism by which this is achieved is yet to be elucidated fully. Several alterations were also identified in the receptor binding domain of the S protein, and these really are of major importance because they are thought to increase transmissibility, but also decrease sensitivity to neutralizing antibodies, and that could be from both natural infection and vaccination. Because of this, a number of the vaccines have reduced efficacy against SARS-CoV-2 isolates carrying these alterations. And this really suggests that a single vaccine will not be sufficient for long-term immunity, and that vaccine reformulation may be required periodically to target the circulating strains at any given point. So Pfizer and Moderna are among the companies that have decided to adopt this reformulation approach for their SARS-CoV-2 vaccines. So that this seems to be the way the vaccine approach is going to go from now on. And although extraordinary developments have been made in the last 15 months with SARS-CoV-2, it's still a relatively new virus. Therefore, further research is definitely warranted to fully understand the effects of all the amino acid alterations that have been identified, and then also to uncover additional or more suitable vaccine targets. So that's all. Thanks for listening. And then I just also want to acknowledge my supervisor, Tony, for all his help, and then the GISAID initiative, which stored all the SARS-CoV-2 genomes, and the UCD National Virus Reference Laboratory. So thank you.
Thank you very much, Fiona. That's absolutely excellent. Talk about being at the cutting edge of what's going on and affecting all of our lives at the minute. So thank you very much for sharing.